Okay, so welcome to this next video in the playlist on antibiotics and antibiotic resistance. Okay, so in this video what we're going to do is we're going to look at a topic known as bacterial charity, and this is a very new phenomenon that we didn't really know anything about until recently. And if you want to know more about this, I would advise that you Google that term, bacterial charity, and you can read the primary research literature if you must. So bacterial charity, and this phenomenon has been so far seen, mainly the archetypal example, the original experiment that demonstrated it was Norfloxacin resistance in Escherichia Coli, so in E. coli. Oh dear, what's happening here? Norfloxacin, Norfloxacin. Is that how you spell Norfloxacin? Yes, that is how you spell Norfloxacin. Norfloxacin resistance in E. coli, basically. Okay, right. So we're going to. I'm going to describe exactly how this works, basically. So let's say we have a colony of Escherichia coli. Right, so here is our colony of Escherichia coli. Okay, so on this plate we have grown some E. coli, basically. Okay, now what we're going to do is we're going to expose our E. coli bacteria to a low dose of this antibiotic Norfloxacin. Okay, so we're not going to expose it to a high enough concentration to kill the bacteria, basically. Instead, we're going to put them in a concentration of Norfloxacin that is going to be great enough to stop protein synthesis within those bacteria, but not to kill them. So here are our E. coli on this agar plate, basically. And now what we need to do is we need to have a brief discussion of how the antibiotic Norfloxacin works. Okay, so basically, when you want to transcribe a gene, what happens is the RNA polymerase enzyme will come to the DNA, and basically it will linearize the DNA in order to transcribe it. So let me explain what that means. So if we use two lines to represent our two DNA strands, so we're going to use this to represent our two DNA strands, and these two DNA strands are complementary, basically. Now, in the bacterial cell, DNA does not exist in this form. You do not just have the two DNA strands linear like this. Instead, what you have is you have them coiled around one another, basically. So, you might twist this one this way and twist this one this way and create coils. So let's imagine doing this. Let's imagine we fix this point and then we're twisting those like that. So we've fixed here and we've fixed here, and what we're going to get is we're going to get this one here. Well, this one is going to go this way, and it's going to come over like this and like this and like this and like this. And this one here is going to go, we're pulling it out, so it's going to go out this way. Okay? Like so. So basically, in our DNA, we have these twists, basically. The two strands are twisted around. It's like taking two pieces of string, and then instead of having them linearly next to each other like this, twisting them round one another. And that's basically the structure, the way DNA is oriented in its native structure. Okay, right. When a RNA polymerase enzyme comes to act on DNA, it can't work on DNA when it's in this twisted structure. So let's say it has a piece of DNA here, and this RNA polymerase enzyme wants to come and work on this piece of DNA. So let's say this is our RNA polymerase enzyme here. Okay, well, when this RNA polymerase enzyme comes to work on the piece of DNA, what it has to do is it has to untwist the piece of DNA which it wants to work on. So let's say it wants to work on this piece, then what it will do basically is it will linearize that piece of DNA, like so. But what it just does is it basically pushes the twist down, so it doesn't actually reduce the number of twists in the overall DNA strand. Instead, it just pushes the DNA twists away, basically, and they end up all sort of squashed in down here now. So you've got this very sort of twisted DNA at the bottom here. Now, when you've got very twisted DNA twisted too much, that is called positive supercoiling. So this is positively supercoiled DNA down here. Okay, and this is dangerous for the DNA, because if you supercoil it too much, then what you end up doing is breaking that DNA, basically, or probably before you actually break the DNA, what will happen is the RNA polymerase won't be able to go any further. So the RNA polymerase will keep going and pushing these twists down, but eventually it will get too tight, and then the RNA polymerase will just be forced to stop. So before the DNA breaks, the RNA polymerase will just be forced to stop. But the point is, if you let this go, if you just let RNA polymerase start pushing the twists down and doing absolutely nothing about this building positive supercoiling down here, then eventually the RNA polymerase would have to stop working, and therefore it would halt transcription too early. So if this was allowed to happen, it would stop transcription from being successful, and therefore you'd stop protein synthesis, because the mRNA needs to be made in order to be translated into protein. So, if you don't deal with this crisis, then you're going to get problems with protein synthesis. Now, the way in which we deal with this crisis is that we have enzymes known as DNA Topoisomerases. And in this video I'm just going to talk about type 2 DNA Topoisomerases, because those are the ones which Norfloxacin acts on. So we're going to talk about type 2 DNA Topoisomerases. Okay, so basically type 2 DNA Topoisomerases, they are designed to remove this supercoiling. So let's say we have a piece of DNA which is too coiled, basically, like so. So it's got too many twists in these two DNA strands that are complementary here. Okay, well what happens is a DNA topoisomerase enzyme, this type 2 DNA topoisomerase enzyme, it comes to this DNA which is too coiled, basically, and it is going to cut both strands of the DNA, and what it will do is it will form a complex, a temporary complex with that DNA. So it's going to cut this DNA into two pieces, like so. So it'll produce double strand cuts in the DNA. Okay, so let's say it's going to cut, where have I gone to? Let's say it's going to cut along this plane here. So here is this piece of DNA from above that point. Here is the type 2 DNA topoisomerase now in the DNA, basically. And here is the DNA going on afterwards, basically, like that. Okay, so it hasn't done anything about the coiling net. All it's done is chopped through both DNA strands and has now formed this complex here where it's inserted and is holding the two sides of the DNA. Now what it's going to do is it's going to untwist the DNA, basically. So it's going to twist around and remove a single twist from the DNA. Okay, so what will happen is it maybe will go to something that looks like this. Okay, so it's removed one of the twists here, so an entire twist like that has been removed. And here is now the type 2 topoisomerase, and below is still the same because it's only removed one twist. Okay, right, so it's now removed the twist, and then what will need to happen is it will now need to dissociate from the DNA. So it will need to religate the DNA together and dissociate. So it needs to religate the DNA together like so, and then dissociate off. So it needs to cleave off. Okay, and then the overall result of this is that it has uncoiled the DNA, and that's a way of dealing with this positive supercoiling that is happening because of the RNA polymerase progressing along the DNA. So as RNA polymerase progresses along the DNA, you'll have these DNA topoisomerases unwinding this positively supercoiled bit here so that the RNA polymerase can continue to progress. And if that doesn't happen, the RNA polymerase will be halted and transcription will be terminated too early, and you'll mess around with protein synthesis, and protein synthesis will stop, basically. Okay, so there are two very special examples of DNA type 2 topoisomerases which are inhibited by fluoroquinolones, of which norfloxacin is a second generation example. Okay, right, and the two enzymes which are both type 2 DNA topoisomerases are, the archetypal one is DNA gyrase. This one is the one that we've known about for a while. We've known that fluoroquinolones act on DNA gyrase. DNA gyrase has another name which is quite confusing. It's called the type 2 DNA topoisomerase, and then it's the second one in this family. So let me emphasize this. The type 2 DNA topoisomerases are a huge, great family of enzymes. It's not just one enzyme. There are absolutely loads of enzymes that are counted as type 2 DNA topoisomerases. And the feature that you have to be a type 2 DNA topoisomerase is that you have to cut both strands of the DNA when you unwind. In order to produce this unwinding of the DNA, you cut both strands. If that's what you do, then you are a type 2 DNA topoisomerase. Type 1 DNA topoisomerases, which we haven't discussed in this video because norfloxacin does nothing to them, they just cut a single strand. So they cut a single strand and then they loop that round the other strand, which was still intact. Okay, so type 2 DNA topoisomerases is a massive, great family of enzymes which affect the coiling of DNA and can either, well, some of the ones we're looking at are ones which uncoil the DNA, i.e. their linker number, which is how many twists they remove, how many coils they remove, is -1. So they remove one twist, and therefore their linker number is -1. Okay, whereas some topoisomerases will have positive linker numbers, i.e. they will add twists in if the DNA is undercoiled, because of course once RNA polymerase ends this transcription process, what will happen is you've removed all these twists from the DNA. So when it all goes back to the way it was originally, you'll have fewer twists in the DNA, and therefore it will be negatively supercoiled. The opposite problem, you'll have too few coils basically, and you'll have to add them back in, and that's the job of the other topoisomerases, which are going to have positive linker numbers. Also, the linker numbers aren't necessarily plus or -1. Some of them will have +2, -2, etc. So they can add or remove more than one twist. The archetypal example are these ones which remove a single twist, and DNA gyrase is an example of this. Okay, so DNA gyrase, or type 2 DNA topoisomerase 2, and I was just explaining why this name is confusing, because there's two of these 2's in here. This 2 up here, this type 2 here, refers to the fact that it is in the family of type 2 DNA topoisomerases, i.e. that it cuts both strands of the DNA when it alters the twist of the DNA. And this 2 here refers to the fact that it's the second member of that family, basically. Okay, and the other important example of a type 2 DNA topoisomerase is the type 2 DNA topoisomerase 4. So this is the fourth member of this family. Okay, now both of these enzymes, as far as we're concerned, do exactly the same thing, i.e. this mechanism is the same for both of them. All that's different is they are slightly different proteins and they have different names. Okay, as far as we're concerned, they're both going to do the same thing. So, what does norfloxacin now do? Well, norfloxacin and all other fluoroquinolones such as ciprofloxacin, levofloxacin, gatifloxacin, nalidixic acid, which isn't a fluoroquinolone but a quinolone, all of these, what they do is they bind to this enzyme, which is either DNA gyrase or type 2 DNA topoisomerase 4, and basically they don't stop it from cutting the DNA. So let's draw this enzyme here. Let's say this is the type 2 DNA topoisomerase 4 or the DNA gyrase enzyme. So I'll colour it in so that everyone knows where we are. So this is now coloured in green. So this is the type 2 DNA topoisomerase enzyme, and now we're specifically talking about either DNA gyrase or the fourth member of this family. So there are others basically which aren't affected by fluoroquinolones. Okay, so this thing that I've drawn on the side then is the norfloxacin molecule, or it could be a more general fluoroquinolone. So this is norfloxacin here, and I can have another chance at spelling norfloxacin now. So here's norfloxacin, floxacin. Right, okay, so norfloxacin binds to the DNA gyrase enzyme, or the type 2 DNA topoisomerase 4, and basically it doesn't stop the enzyme from cutting the DNA. So if we draw this mechanism again, then what's going to happen is it's still going to cut the DNA. So what's going to happen is you're going to cut this DNA, okay, so the enzyme's going to insert in here, okay, with the fluoroquinolone or the norfloxacin bound, like so, and the DNA is still the same underneath, so everything's going well so far, what will happen is it will remove the twist again, so it will still remove the twist. I will remove the positive supercoiling, so here's the norfloxacin drug. So what will happen is you will remove a twist, so you'll go down to this. Here's your DNA type 2 topoisomerase, which is either DNA gyrase or type 2 DNA topoisomerase 4. Where are we? There, okay, to there. And now comes the problem. Basically, it removes the extra twist, but now, when it comes to actually religating the two DNA strands together, that doesn't happen. Something about the norfloxacin seems to stop the religation of the DNA, and therefore leads to an accumulation of these complexes in the DNA. Okay, so it therefore inactivates these enzymes because they're just going to sit in these complexes now. And if you give it at a high enough dose, what can happen is you can get so many of these complexes in the DNA of the E. coli or the other bacterial cell that DNA repair mechanisms are activated so much that the cell says, actually, no, we've got too much of a genetic crisis here. We are at risk of causing havoc, basically. We're at risk of getting some horrendous mutation, and therefore we'll activate apoptosis and end this, basically. Okay, so if you give norfloxacin at a high enough dose, it induces the E. coli cells into apoptosis, or the bacterial cells into apoptosis. We don't want that. We're going to give it at a slightly lower dose, where you just get a few of these complexes forming, basically. And you inhibit, therefore, the DNA gyrase, or the type 2 DNA topoisomerase 4 somewhat, because the enzymes are sitting in the DNA in these complexes, basically, so they're not performing their function because they're stuck, basically. So, what's that going to mean? That's going to mean that the E. coli is not capable of protein synthesis, because now when it tries to transcribe genes, RNA polymerase is going to work, it's going to build up this positive supercoiling, but these enzymes aren't functioning anymore, so they're not going to be able to remove the positive supercoiling. So, RNA polymerase is going to just say, oh, well, we're going to have to stop. It will terminate the transcription too early, therefore you won't get the full mRNA made, and therefore you won't get the full protein made, so it's going to stop protein synthesis. Okay, so if you don't have protein synthesis, what happens to you, basically? Well, you don't need protein synthesis to live, The bacterial cell has all of its proteins already made, so it can live quite happily with the proteins that it's got. However, if a bacterial cell is to divide, like so, then it's going to need to double the amount of protein it has, because, you know, it's got to have the same amount of protein that both cells would have. So it would have to have protein synthesis. And if we've given a dose of norfloxacin that stops protein synthesis, then it's not going to be able to divide. So at lower doses, norfloxacin acts as a bacteriostatic antibiotic rather than as a bactericidal antibiotic. So at high enough doses it will actually be bactericidal, but at lower doses it will be bacteriostatic. And there is a concept known as the MIC, or the minimal inhibitory concentration, which basically means the concentration of drug that you need to have the bacterial colony in order to stop it from dividing, basically. So the minimal inhibitory concentration is the concentration of drug needed to stop the bacteria from dividing, and basically usually this is too low to actually kill the bacteria, but it will stop the bacteria from dividing. And this is a concept that goes through many antibiotics. The distinction between bacteriostatic and bactericidal antibiotics is useful, but mainly most antibiotics, if you give them at high enough doses, will become bactericidal, and at lower doses they'll be bacteriostatic. So the minimal inhibitory concentration, this concentration that you need in order to get bacteriostatic effect, that's a concept that goes over many different antibiotics, but here I hope it's clear that it's the concentration we need to put in to our agar plate here in order to stop the E. coli from synthesizing proteins and therefore stop them from dividing, but it's not high enough to actually kill the cells that are already there. It's not the it's not the bactericidal concentration. Okay, and we'll continue this discussion in the next video.